Right. Hello, everyone. Yes, can you hear me? Yes, you can. I'll hold it a little bit further away from my mouth. It's great to see so many people here this morning for this talk that we have as part of the HEAS series on human evolution and the Paleolithic with Emma Loftus from Ludwig Maximilians University in Munich. Emma is originally from South Africa. She did her undergraduate degree at the University of Cape Town. And following that, in 2011, she went to the University of Oxford, where she worked as a master student on the Master of Archaeological Sciences there, which is where I believe we probably first met. Following that, she did a PhD, or a DPhil, as I should say, in stable isotope geochemistry, which is her specialist area of expertise, working on material from South Africa, from the Middle Stone Age, on these amazing South African sites, looking at sea surface temperatures and Paleolithic times. And that work was supervised by Julia Lee Thorpe and Judith Seeley, I believe, as well. Two big names of the isotope world. Following that, she held she's held postdoctoral positions at the University of Cambridge, where she was a Leverhulme Early Career Trust Fellow, which is a very prestigious grant to get. And following that, she got a Marie Sklodowska-Curie Award, which she took to the university in Munich, where she's now based. So now that we've got everyone in the room, I'm really thrilled to hand over for this talk to Emma. There'll be some questions afterwards. If you're online and you want to ask some questions, you know what to do. Just send something in the chat and we'll include your questions at the end. Emma, over to you.
Thanks, Tom. Thank you so much, everyone, for this introduction to come talk to you today. I've been watching you guys for the last few years, seeing what everyone's been up to and really wanting to come and check it out. So it's a real treat for me to be here. I'm currently based just over the hill in Munich, where I've been for about the last 4 years. And as Tom said, prior to this, when I was based in the UK, I was working on the archaeology of Pleistocene and Holocene hunter-gatherers in Southern Africa. But since moving to Munich, I've kind of pivoted to working on farming communities, and that's what I'm going to talk about today. Okay, so I am going to take the liberty, since I'm here, to talk to you about two quite different types of projects that I've been working on. Firstly, I've been working with radiocarbon dates. I thought that might be a little more interesting to this crowd. And I'm particularly working with these large data sets of dates, and in this particular case, working with dates from farming communities. Secondly, then I'm going to talk to you a little bit about the funded projects that I've had, which is working on lipid residue analyses of farmer-made pottery. And then I'm hoping to kind of bring it together at the end with a few slides. Okay, so to set the scene a little bit here, a few years ago, I published a database of archaeological radiocarbon dates from Southern Africa. So it's hosted, it's online, it's hosted by the Oxford Lab. But the key thing I kind of want to point out is that it's really small. So at the time, we had about 2,500 dates, for 50,000 years. It's quite a long time period. And most of those actually fall within the last couple of thousand years. This was quite a fun project for me, sort of at the late stage of my PhD, because I started to read and think about these methods that are out there for aggregating radiocarbon dates. But what was pretty clear was that we weren't going to be able to throw some of these really high-powered methods at such a small data set over these really long timeframes. So it was, we weren't going to be able to tackle these questions of demography and landscape use and niche sizes and things like that people have been able to work on with these kinds of methods. And we would need to define our questions at maybe smaller scales. The second paper that kind of leads up to what I want to talk today, this came out, I think, last year. And this is just a sort of catalog of directly dated human remains from Southern Africa. So those hosted in national repositories. And this was a really straightforward paper, just did some really basic kind of modeling. So you can see there the kind of split between, the spatial split between burials related to foraging or pastoralist groups, what we call in Southern Africa the later Stone Age, or those related to farming communities, what we call the Iron Age. And again, the key thing I just want to point out about this is the pretty small sample sizes that we have in the region. But again, the other point to note is the discrepancy between the sample sizes for foraging people and those for farming people in the region. Okay, so this is that data set. This is what appeared in that paper. So this is a really simple kind of conservative aggregation tool, kernel density estimation. And you can see the curve for foragers in red and that for farmers in green. And so the whole data set for foragers spans about 10,000 years, but actually most of the data sets of remains come within, let's say, the last 3,000 years. And the other thing to kind of point out here is that there are some really marked peaks and troughs in this kind of curve, right? And so I'd done some work thinking about what these kinds of these fluctuations might mean. And the year before, actually, I'd published a paper with Susan Pfeiffer working on that big peak at about 2000. And we had suggested that related to a change in forager burial behaviors at the arrival of new people on the landscape about 2,000 years ago. So the arrival of pastoralists. So that's kind of a Western Cape peak there. And then that peak there in the mid-Holocene, I think we can interpret that. I haven't written about this, but I think we can interpret that in terms of some dynamics that are happening in the Southern Cape in the mid-Holocene. So the kind of point here is that this big curve, this red curve, it looks broadly correct. We can kind of interpret it in terms of things we already know about the archaeological record of foraging people. And there's probably some potential, as I kind of shown in the Western Cape, there's some potential to unpack that at smaller scales, at regional scales, temporally, things like that. So even though this is quite a small collection of direct dates, less than 500 dates, it does look broadly correct. And we can address some some questions about forager behavior. Does the same hold true for the farmer record? Where we only have about 65 direct dates for 1,500 years. What kind of explains the shape, this bimodal shape of this curve? Is it something about a change in mortuary behaviors through time? Is there something demographic going on? Well, pretty obvious, pretty immediately, it was clear that there was a couple of features of this distribution that didn't look right. So firstly, the oldest dated settlements, so the oldest radiocarbon dates that we have for farming settlements, actually date to within the first part of the first millennium. So quite a bit before the earliest dated burials. But the bigger problem was this big dip in the first part of the second millennium here. Because that overlaps with a period known in South Africa as the Middle Iron Age. So the Middle Iron Age is this period of pretty intense settlement with quite high archaeological visibility. It really refers to this region mostly in the northern part of South Africa, so along the border with Zimbabwe and Botswana, the so-called Shashi-Limpopo area. And it characterizes a period of state development. So we get the emergence of a society ruled by a royal elite, centered on this urban capital shown in the picture here, at Mapungubwe. So Mapungubwe is today a World Heritage Site. And crucially, we knew that many, many burials, human remains, had been recovered from Middle Iron Age sites, including something like 100 from a single site alone, K2, which is a precursor to Mapungubwe. So to clarify, we knew that there were many times that number of human remains from farmer contexts, but relatively few of them have been directly dated. And so I think that this distribution is just kind of a poor one because of bad sample numbers. And there's a number of reasons why we might have fewer dates for farmers as for foragers. So one thing is collagen preservation is generally very, very poor across Southern Africa, and especially in the regions where farmers were settled. Another is that for these types of archaeological contexts, there are maybe better, more precise dating methods available. So there are these quite well worked out ceramic typologies, for example. We have things like glass beads can be found in the site. These might be better than radiocarbon in these time periods. the other thing I think is that within the last couple of decades, at least, although there's loads of research in this type of archaeology, lots of interesting questions, none of these questions was particularly chronologically focused, and so there wasn't a lot of urgency about trying to develop radiocarbon chronologies in this archaeology. But I think the biggest reason for why there are so few direct dates is because of the sensitivity of dating human remains. Of course, there's been a growing recognition sort of globally in recent years about the ethical issues of sampling human remains. And this is, a global trend. But in South Africa, there are specific kind of themes and emphases that have been ongoing for quite a long time now. So now, of course, there's these sensitivities, but even earlier, there was issues around directly dating human remains from these types of contexts, from the archaeology of Black farming communities. So, for example, under the apartheid era, archaeologists had to be quite careful working on these questions about the longevity, the deep time nature of Black farming occupation on the landscape. And then in recent years, with regard to the Iron Age record, so this type of archaeology that I'm talking about, there's been a lot of broader kind of community political support for the return of these human remains to the communities that live on the landscape now, and their reburial, as shown in these pictures. And certainly, I think within the last few years, archaeologists tend to avoid having to excavate human remains where that's possible, where it's not necessary. So consequently, because of all of these kinds of issues, I think that data set of 65 radiocarbon dates is unlikely to grow very much. I think that's kind of what we've got. So can we improve on this curve? So I worked on this problem with Marlies Lombard and Marina Steen, who's a bioanthropologist. And we had this data set of human remains that are hosted in South African repositories, so 10 institutional repositories. These are museums, universities, and include some reburial facilities as well. And we also knew that oftentimes these individual human remains, there was quite good age information associated with them, if not a direct date. How could we incorporate these? Okay, so I popped all of my dates into a kind of dating triage, all of the age information that I had into dating triage. And we could categorize the age information into one of these kind of three categories. So the first and best, of course, is where we have a direct date on the set of human remains itself, or one in very, very close association. So another individual in the same grave pit, for example, or a piece of charcoal that came from the grave pit. The next is the largest group, is where we can simulate a date based on a modeled age for the site. And fortunately, many Iron Age sites have quite short occupations, so over the course of perhaps several decades or a couple of centuries. And OxCal can simulate a date which we can then input similarly to a regular radiocarbon date into our models. Finally, we have about 70 sets of remains where the age information was much broader. It could be, over several hundreds of years, or I was much less sure about the association between the dates and the human remains. So perhaps some of these sites were excavated a very, very long time ago. Some of the information isn't so clear. And so these were kind of the poor quality data. So then what we do is we just model these three overlapping data sets. At the top, and that's broadly what we presented in that original paper. The scale is very compressed, but it's broadly that same bimodal distribution. And then we add in the second model, in the middle of the screen, we add in those medium quality that age information. And you can see that this is completely different distribution now. Finally, when we add in, so I had kind of expected that when I added in the poor quality data, everything would get kind of smeared out and wouldn't look so interesting anymore. But actually, if anything, the whole pattern becomes kind of exaggerated. And so it kind of suggests to me that poor quality data is actually broadly consistent with the medium quality data. And this new curve, if we take all of the data that we've got, this looks a lot better. We've got this major peak here centered on about 1200 AD with this really sharp decline at about 1400 AD. And this to us clearly reflects that really large number of burials that were recovered from the sites in the Shashi-Limpopo region. Okay, so what have we got? We've got a wiggly line that now looks a lot like what we already knew was the case. The point, though, now is that we can start to unpack these data sets and start comparing subsets of the data to try and understand what might be driving the peaks and troughs. So the immediate issue is that we're kind of a victim here of our own success, and we've got this huge blue bump, which may be obscuring other features of the distribution. So we can compare, if we take that, so it's shown in blue there is the original distribution, and if we take out dates from just 3 sites, so these are 3 sites in the Middle Iron Age landscape, K2, Mapungubwe, and Schroeder, we see something really, really interesting. So firstly, we can see how that landscape, that small landscape, really, really dominated the Iron Age world of Southern Africa at the time, because there are almost no dates, burial dates from that anywhere else at that time period. The other thing is that now the green curve, so excluding those 3 sites, has quite a lot more structure in it, including some interesting peaks that were formerly obscured. So for example, that peak towards the end of the big blue bump in the green, that was formerly hidden inside it. And actually, if I look at those in more detail, if we unpack what that smaller green bump is, those are burials that are from a landscape to the south of Shashi-Limpopo area. And so we've kind of interpreted this as reflecting migration dynamics at the collapse of the Mapungubwe polity. So we can do stuff like that. How do we explain the big blue bump? What does it actually reflect? Is it a shift in burial behaviors, so mortuary behaviors? Or is it something kind of more straightforward, like a demographic response? And so people who work on these methods, you know that interpreting something like demography from radiocarbon dates is really, really difficult. There's a lot of assumptions we have to make. But I actually think that in this case, it is a demographic response. So unlike that Western Cape record where it was a change in mortuary behaviors, we think related to the arrival of new people on the landscape, here I think it's a more straightforward demographic response. And one of the lines of evidence that supports that is the composition of the individuals, so the demographics of the individuals from, for example, those 3 sites in the Middle Iron Age period. So shown in purple there, large numbers of these individuals are juveniles or infants. And so this kind of death assemblage, as it were, we don't have any other evidence for like a plague or anything like that. This is very suggestive of a rapidly growing population. We can also unpack this at spatial scales. So this is work that I did with Brian Chase. We split the data set here into 3. One down here in the southeast, mostly in KwaZulu-Natal, up in the northeast, which includes that Middle Iron Age landscape, and then over in the northwest. And the points, these 3 groups actually make sense from an environmental perspective. There's a moisture gradient across this landscape that maps onto this here. There's an escarpment that's running there. The problem, of course, that we have with this is we can't split things up too much. So we start to get these really small, unsatisfactory data sets if we go too far. And I think arguably the KwaZulu-Natal southeast record down there in yellow is already starting to get a little bit too small. Okay, so if we look at these are again the OxCal models for those 3 regions now. And overlain on that is the timing for the medieval climatic anomaly and the Little Iron Age locally. And the sort of pattern that jumps out at everyone, I think, immediately is that kind of mirrored patterning between the northeast region and the northwest region that sort of lines up with the medieval climatic anomaly. And then what we did was, of course, to compare this with regionally relevant paleoenvironmental records. And along the bottom here, you can see is a measure of El Nino activity, which indicates that middle Iron Age peak in the northeast really coincides with a period of reduced El Nino activity in this region. In this region, what that translates to in terms of the environment is higher and more predictable moisture availability. Okay, so this then we could interpret in terms of some ideas that were already out there. It aligns very nicely with paleo-environmental and archaeological evidence that suggests that this period of the Middle Iron Age was one of improved agricultural conditions in the Shashi Limpopo. So today, this is not a very productive landscape, but at the time, potentially there would have been much greater agricultural productivity alongside this large river due to seasonal flooding, which would have facilitated these large grasslands. The grasslands, of course, are good for cows, so for cattle and things like that, but they're also really good for elephants. And the Middle Iron Age is also known for the establishment of these trade links into the Indian Ocean world. One of the major goods, of course, from this region being worked ivory. So it kind of is a nice little story. Okay, pivot now to this, what to me is a sort of new line of research working on lipid residues. All of the work I've done on this so far has been over in the southeast part of South Africa, so KwaZulu-Natal. Most of what I've worked on is the so-called first millennium farming communities, or the early Iron Age, which is the first millennium CE. The other point, I've got data from 5 sites that I want to show you. But the other point here is that this landscape was, of course, at the time also occupied by hunter-gatherers. The big themes here are about this, huge migration of people that took place, the farming people that took place starting over in West Africa, moving over gradually over several thousand years, ending up in Southern Africa about 2,000 years ago. And this map, I think it's very useful for kind of highlighting a couple of points, kind of some background ideas. It's based on ceramic typologies. And so I think it emphasizes, firstly, the kind of lack of detail that we have about the exact migration routes as people were moving south. There's still a lot that we don't know about exactly how these groups related to one another through time, how they made a living, things like that. The other thing that these kinds of maps do, of course, is that they imply this really deep continuity through time. So there's I would say, a general kind of acceptance of the idea that the cultural traits in more recent periods, so within the last 1,000 years or so, have these really deep roots going back in time. And so, for example, that we could use ethnographic observations from the historical period to interpret the archaeological record. The other thing that's kind of reflected here is that the movement of these societies proceeded as a kind of cultural package. So the technologies of ironworking, ceramics, and different agricultural products all kind of moved together. And of course, this has been challenged from different directions, but this is a broad kind of understanding. So I had some more focused research questions, of course. This was work done as the Marie Curie. And so the groups who settled this region in the 2nd millennium, the so-called later Iron Age groups of people, who are ancestral to modern Nguni language groups in Southern Africa, had cattle-centered societies. So cattle were really, really important for their economies, but also in terms of mediating social and political relationships. And from what I thought was really interesting is that from an ecological point of view, cattle, of course, can be really transformative agents on the landscape. So it had been suggested in this region of KwaZulu-Natal that cattle had been responsible sometime in the late Holocene for the transformation from wooded environments to these grassy rolling hills that you get in the area today. But there was a lot of debate actually within the zoo archaeological literature about whether these first millennium groups actually had cattle at all. And we've got quite poor bone preservation, these really fragmentary assemblages. And so it was possible to argue this question in different ways. So the question for me was, could we detect dairy in the ceramic residues? And what kind of dairy? Can we distinguish them between cattle or sheep and goat? The second question concerns marine resource use. So there had been this for decades. There have been the suggestions that these pioneer farming groups, as they moved down into southern Africa, this is quite a different set of ecosystems to where they had come from. They had managed to move quite quickly by following the coastline, and that they had used these rich coastal resources like shellfish to facilitate and to buffer their subsistence as they were, transforming the landscape and establishing agricultural systems. Again, though, there wasn't actually all that much evidence for this theory. It's quite nice, appealing theory, but in fact, the number of sites is quite small. So we do have some early radiocarbon dated sites from the coastlines, but there's only a few of them. And many of them haven't been excavated to a very high standard or properly radiocarbon dated. So we've got only a handful of dates. It also contrasts very strongly with our understanding of subsistence in the second millennium, where later Iron Age farming people really, these are not maritime cultures. They are not fishing cultures. They don't access a lot of resources from the sea. And if anything, there's actually kind of a prohibition on eating fishy foods. So could we detect something in the ceramics? And then, of course, there's loads else that you can do with lipid residues, all sorts of fun plant foods that we could maybe find. We could maybe find other kinds of aquatic foods. Insects, of course, are eaten widely across Africa, at least until the recent past. And insect foods like honey, all sorts of fun bonus stuff. Okay, very brief introduction to the sites. 2 of my sites were coastal. Those are the oldest dated sites. And 3 then were from the inland, including this multi-component site, Kwaganda. And what I should say is that I managed to span with the samples that I selected the entirety of the early Iron Age. So going from really the very beginning to the very end. The other point, though, is that all of these were excavated a really long time ago. So excavated from the 1970s through to the 1990s. And this is just a shot inside one of the boxes. And you can see everything's, archaeologists, lots of touchy. They've been painted on. They've tried to reconstruct them and stuff. They had been heavily handled. And we thought that this, we didn't know what this would mean for trying to recover dietary lipids. Very brief overview of my methods. I was really lucky that I was working with a brand new archaeological lab that I essentially had to myself. For Simon Hammann, he's now moved to Stuttgart. And we were working with the acidified methanol extraction protocol mostly. And so within the last decade or so, this method has been responsible now for these papers that you see where you get these really huge numbers of samples processed. And even though I was just learning the method, I was able to get something like 200 archaeological samples processed, these really rich kind of data sets. And then this is the instrumental methods that we used. So analyzing the lipids using GCMS and a little bit of isotope work to try and do some further discrimination. Okay, so these pie charts show the success rates for my different sites, and blue is good here. So this is a kind of widely used metric for whether we've got adequate amounts of lipids to kind of interpret them. Is 5 micrograms per gram of ceramic sherd. And this was really, really positive. Remember, bone preservation is not great in this region, and so we were sort of unsure what we would get, but it looks like lipids are generally very positive. Oh no. Okay. Yes. So sorry, I should have come this far down. Okay, so this was trying to sort of, so chromatograms are really boring to look at, so I'm going to try not to show you too many of them. And so this plot moving from, okay, I'll just have to kind of summarize it here. So essentially, I got a range of different kinds of recovery rates across the different contexts. Some of them were pretty average, kind of meh. Some of them were exceptionally juicy. So these really rich fatty sherds, full of, in particular, animal fats. So these, this really kind of diagnostic duo of fatty acids, C16 and C18, or steric and palmitic fatty acids, which are very indicative of animal fats. The other thing that I have in pretty much every single sherd was phthalates. So these are contaminants, plastic contaminants that were probably acquired somewhere during the curation process. But really, fortunately, the peaks are super diagnostic. They're quite clear. They don't massively interfere with the dietary lipids. And so they're there, but they don't really mess up our interpretation of the dietary stuff. I also had lots of planty kinds of signals of different levels throughout. And then a couple of things that I want to talk about just because they're interesting. So the first one that was pretty worrying actually was DDT. So this recovered from one site only that is located in South Africa's oldest reserve, wildlife reserve, Isimangaliso, what used to be known as St. Lucia. And of course, DDT is this chemical that's used to combat insect-borne diseases. And it was used in vast quantities in Southern Africa in the 1960s and 70s. It was banned as in the rest of the world in the 1970s, but it was actually used for quite a bit longer unofficially by farmers. And I should say here that experimental studies within lipid residue studies for archaeology have given us broad confidence that this isn't generally a massive problem, that the sherds don't really seem to reflect signals from the soils, from the sedimentary matrix. But in this case, of course, DDT is obviously this really toxic, persistent, damaging compound. And what's more is that it has been found to be completely pervasive. So this study here of crocodiles from this ecosystem found that they had some of the highest concentrations of DDT from wild tissues ever recorded anywhere. So this is obviously just completely pervasive through this ecosystem, even though these sherds were excavated in the late 70s, right? So I don't think this is a broader problem for interpreting archaeological lipids, but it's obviously kind of interesting and worrying for this environment. In terms of the archaeology, one fascinating find is this example of beeswax or honey. We can't discriminate between them, so let's say honey. And this comes from one of my oldest dated assemblages from Mzanjani coastal site. And this really excited the archaeologist that I'm working with, Gavin Whitelaw, because honey is proposed as this exchange item with hunter-gatherers. So we have lots of evidence. This is forager rock art here. We have lots of evidence that honey was important, not just from a dietary perspective, but also sort of had this kind of symbolic importance for forager communities. And we know that in later periods, foragers certainly were providing or supplying farming societies with wild foods, including honey. And what's kind of interesting as well about this particular sample is that it's super, super pure. So it's not mixed in with anything else. So this was potentially a storage vessel or something like that. Another thing that we have is ricinoleic acid or ricin. The possible sources for this, of course, are the classic is the castor oil plant. We're not sure that this is what this was in this region because it's not believed to be native to Southern Africa, further north, potentially more common. But there are other plants that would have produced this kind of compound. And of course, it's widely known for medicinal usage. And what's interesting is that this has now been recorded from a number of different types of contexts in Southern Africa, including a so-called poison applicator stick from Border Cave at about 24,000 years old. Another, an arrowhead embedded inside a bovid femur. And then also from an early Iron Age context, the one other study on early Iron Age ceramics that's been done in the region, slightly to the north of where I'm working. Okay, so very briefly to talk about isotopes. The isotope, if we directly measure the carbon isotopes of these two fatty acids, C16 and C18, that helps us to discriminate the types of animal tissues that we're interested in. And I'm obviously particularly interested in trying to detect dairy. So this is because of differences in how these fatty acids are synthesized by animals. So for adipose tissues, animals have to synthesize both fatty acids de novo, so they are both fractionated to the same degree. Whereas for dairy, animals only synthesize C16 de novo, and they're actually grabbing C18 from diet, which in this case, of course, is plants. So the C18 is not fractionated. And so we get this difference of a couple per mil. This is a kind of standardized plot used to display these types of results. And so you can see, depending on where you fall on the y-axis, discriminating between dairy or adipose. And then on the x-axis, we can also get an insight into the extent of C3 versus C4 dietary inputs. And in this part of the world, in Southern Africa, grasses are mostly C4. So this is a very helpful tool to discriminate between grazing or mixed feeding animals and browsing animals. Sorry, browsing and mixed feeding animals and grazing animals. Right. So I only have a pretty tiny data set of isotopic results. These are quite difficult measurements. But the plot shows a couple of interesting features. So the earlier samples shown in red and green show I've got C3 ruminants mostly falling in this area of the plot, C3 over to perhaps mixed feeding. But with an increasing proportion of C4 through time. And in the final phases, so this is late stage at Kwaganda, the so-called Nshakhan phase, this is where we start to see dairy very clearly in the record. So this indicates overall, I think taken together, a shift from animals that were eating C3 or mixed diets, so perhaps either wild browsers or stock such as goats. Obviously, that's not really, anyway. Moving over time to something like grazers that are only eating grasses, and then dairy grazers, which would be something like cattle. So this is a major finding because it suggests that in the earlier phase, we don't have any evidence for cattle dairy. And that supports these people who think from a zoo archaeological perspective that there isn't a lot of evidence for cattle very early on. Okay, now I kind of want to gesture towards where I'm going to go next. Just a few slides here. Sorry, what am I? Yeah, okay. So this is a kind of approximate chronology that we have for Southern Africa. And it's very much conceived as these two parallel kind of chronologies. So we talk about the chronologies of agriculturalists, the Iron Age, or you talk about the Stone Age for foragers. And archaeologists in the region tend to work on questions related to one or the other. There are some people who've managed to bridge these, but it's quite difficult because of the sort of conceptual frameworks that we have in the region. We, of course, know that farmers and foragers were living alongside one another for centuries, but there hasn't been a lot of work trying to look at this from an integrated kind of perspective. So this is the types of evidence that you can find in the different sites indicating this kind of exchange or contact. And, we know, for example, that foragers were acting as ritual specialists for farmers, so rainmakers, as it were, in some places. But I'm interested in the so-called invisible exchange. So this would be mostly, for example, food products that we no longer find in the archaeological record. Could ceramics be a way into that problem? How did foragers actually use the ceramics that they obtained from farmers that we find in forager sites? Do we see big differences in the lipid assemblages? If we look at this at a kind of assemblage level, do we see big differences between adjacent farming and foraging sites? And, interesting things here would be things like aquatic foods, which aren't really one thing I didn't point out is that I've got no evidence that farmers, even in the earliest periods, were accessing aquatic resources. So things like aquatic foods, things like honey, things like dairy, were foragers able to access dairy in the later period? So I've done I've done a little bit of work starting on this year. Building on that earlier project. So I've got some assemblages now from forager sites that are adjacent to farming sites in this region. I should point out that there are no indications that foragers ever made this pottery. This pottery fits very firmly within farming typologies, and there's no suggestion that they were making it. So they're certainly obtaining it somehow from the farmers. So I've got sites up here in the Tugela River Basin, and then down here in the Ananda River area closer to Durban. And this is just a kind of some very preliminary results. My data sets are quite small as yet, but you can see, again, pretty positive results in terms of data quality. So given that we can sample really large numbers of sherds using this method, we can get adequate kind of sample numbers. And some sites are, of course, better than others. So I want to quickly talk about Mzinyashana. This is a very classic rock shelter site from the Tugela River Basin, excavated by Aaron Maisel with an assemblage, with a set of deposits going back over the late Holocene. It's got a pretty decent radiocarbon chronology for this part of the world, 11 dates. And it's an interesting site because Maisel actually used the site to propose some of his broader theories about the connections between farming and foraging people in the area. In particular, he believed that at this site, there is a forager or hunter-gatherer occupation for most of the period. And then at the top, the very top, he believed that actually farmers came into the site and occupied it in the later period, which is kind of interesting. Okay, chromatograms, but these are quite easy ones to look at. So those two peaks, so we're moving here from the oldest deposits at layer 5 through to layer 2. And these two big peaks that you've got here are those C16 and C18 fatty acids. So that's that very strong animal signal, animal fatty acids, dairy maybe, or probably more likely some kind of meaty product. And so you can see also, this is my internal standard. So these are juicy samples. These are really, really rich in these animal fats. And so kind of the same. We've got, again, a really, really juicy sample here, again in layer 3. And Maisel wasn't too sure about layer 3. He thought maybe it was farmers, but he wasn't exactly sure what was going on. So the transition in occupation happens somewhere there. It's really different in the topmost samples. These are not rich in lipids at all. They've got almost nothing in them. And what they do have is just these kinds of very undiagnostic alkanes and things like that, which we sort of tend to throw our hands up and say, oh, plants of some sort, because they're just not, they're not super diagnostic. We can't do much with them. And so there is a definite shift. And I think this kind of raises lots of interesting questions about who were these farmers who came into the site? How did they make a living? How did they end up here? And in terms of these questions, this is rich ground, I think, in this region to address these kinds of questions using these methods. So lots of mileage here. We've got dozens of these rock shelters that are sort of adjacent to farming sites with farmer ceramics in them. And I want to develop these kinds of comparative data sets. I've been mostly working down here in this very well-watered, we say, very moist kind of landscape of KwaZulu-Natal. But a lot of the sites would have been in much less productive ecosystems further to the north. And so we could, we could plausibly think that these kinds of relationships, the types of risk and that people are willing to accept, is going to be modulated by this kind of environmental variability across the landscape. This is where I'm hoping to go next. Thank you very much.